to sort of go over the outline for today, so I thought I'd give a bit of a background on precision medicine and bioinformatics, because I think we have a very, very diverse audience. And then I'll talk a little bit about the work that we've done. So in particular about the multi-platform clinical pathways and another project on 3 words that you normally don't really see together, lipidomics, chimpanzees, and coronary artery disease. And yeah, more to that when we get there. So to start off with a bit of background, I thought, let's think about how classical medicine is often done. So here's just an example of a cancer treatment. I won't even try to pronounce that. Vaish was trying to teach me how to pronounce it. It did not work. But anyway, so there's this like new treatment, right? And you want to know, is this treatment effective? So what do you do? Well, you run like a clinical trial. You give the treatment to some people, you give a placebo or a baseline treatment to some other people, and you sort of see who has the better outcome. So in the development of this drug, this is sort of the plot from their paper. And here, this is just plotting some survival curves. So on the x-axis, you've got like the time, and then on the y-axis, you've got the overall survival percentage. So the percentage of the cohort that survived at that particular time. And in this bottom curve is the baseline treatment. Like that's this, I don't know what this is, GP 100. So you have this baseline treatment, and then you have these new treatments, and then this new treatment plus the baseline. And you can see that like the survival curve is higher, which means that sort of when people were on the treatment, they generally survived for longer. And so, in the paper, they report, survival goes from 6 months to 10 months. P-value is very low. So it looks like this treatment is really effective. So, treatment's really effective. You put a patent on it. You start prescribing this to patients with particular types of cancer. And then, everyone's happy. Or is everyone actually happy? So if you look at the survival curve, you'll actually notice that there were some people who were on the drug, so like on that top curve, but they still had quite a poor survival outcome, right? And conversely, there are some people who were not on the drug, like on the baseline treatment, and they actually survived for a very long time. So what this means is that people have very different responses to the same treatment, right? And this isn't very surprising, right? Because, people are very diverse, right? People have, different genetics, different environmental factors. People have different comorbidities. And so it's not very surprising that if you give the same treatment to different people, they will respond in very different ways. And so this brings us to the question of, can we study medicine at the individual level? So rather than sort of thinking about, if a treatment is effective on average, can we think about whether a treatment is effective for each individual person? And that brings us to the concept of precision medicine, which is one of the key topics for today. And in fact, this idea has actually gone back for quite a long time, so it's nothing new. So Hippocrates, who's often deemed as the father of modern medicine, has this famous quotation saying that it is far more important to know what person the disease has than what disease the person has. Right, so here it's sort of saying that understanding the individual is more important when it comes to treatment than the disease that they actually have. So precision medicine is a concept that sort of existed for quite a while. So you might be thinking like, so why is it such an interesting thing to study now, right? And also, like, why is a statistician trying to mansplain medicine to a diverse group? And to address that, I'm actually going to mansplain a little bit of biology as well. So firstly, sorry to any biologists in the audience. I'm going to absolutely butcher this, but the way a lot of biology works at the molecular level is that you start off with DNA. So you can think of DNA as like one really, really big molecule that each of your cells has, and this molecule is sort of determined from birth. And then what the cells do to sort of do anything is that using that DNA, it makes these smaller molecules called RNA, And then from that RNA, it then creates proteins, and that's sort of what the cells do to sort of do like every function. Okay, so this is like a rough, really butchered explanation of molecular biology. But the reason why it's so interesting to look at sort of medicine and biology at this day and age is that technology has really advanced in recent years to the point where we can detect and observe these molecules at each of these levels. And so in fact, at these many different levels, we have all sorts of technologies that can detect these. So for example, we have a whole range of genomics technologies, which can measure DNA. We have epigenomics, transcriptomics, proteomics, metabolomics, lipidomics, all of these which we call omics. And in fact, there's many more that I couldn't fit on this slide. And the development of these sorts of omics technologies is often compared to the invention of the microscope. So, back in like the 1600s when microscopes were first invented, it actually led to like a whole revolution in biology because it gave scientists a way of like sort of seeing a whole new world of like cells and microorganisms. And so now that we've sort of developed these omics technologies, you can actually think of these as we now have the technology to observe what is going on at the molecular level, right? And so this gives us like a new perspective on sort of what's happening at that biological level, at the molecular level. And that's really exciting, from a medical point of view, because this means that you can better understand the causes of these sorts of diseases. And these are also potential biomarkers we can use to detect diseases. So it's a really exciting field. And not only are there a lot of these sorts of technologies, but these technologies are also becoming more scalable and more affordable as well. And so here, this is a plot of the cost to sequence the human genome, which is sort of one type of this sort of data. And you can sort of see like from about 25 years ago, the cost to collect this sort of data for one human genome was about $100 million, right? So definitely not very affordable. But over the last couple of decades, you can see that the cost has like really plummeted. And I think now you can even get your genome sequence for like several $100s. And something that I really want to point out is that in this graph, the y-axis is actually logarithmic. So when it's going down, it's actually going down really quickly. And in particular, we have a Moore's law here for comparison. And so what this is actually showing is that the cost of these technologies is decreasing faster than exponential. So the accessibility of these technologies is really growing massively. So this is pretty great, right? So we've got this really accessible technology. We can generate this data, which provides a lot of insight. So why haven't we sort of solved all diseases? And so it's because there are a lot of challenges in this field as well. So for a startup, there is a lot of data. So because this technology is getting more accessible, we can generate these really crazy amounts of data. And in particular, if you look at sort of the genomics level, there's like 3 billion base pairs and 10 million common single nucleotide polymorphisms like gene mutations. And there's about 20,000 genes to look at. And so the scale of the data that we're generating is like, it's got a lot of features in there. And just to give another example, so the Cancer Genome Atlas is like one database, and that database has 2.5 petabytes of data in there. And that's like 2,500 terabytes. So these data sets are really massive. And trying to sort of deal with this data, is quite challenging. And in particular, like at a more sort of local level, in the School of Maths and Stats, we have like some research servers, and yeah, and I feel like every couple of weeks we run into issues with our servers running out of storage space. So I've just got a few screenshots from our Slack channel, and this is all from this year. So just in the last few months, yeah, you can see we have a lot of issues with this. Oh, sorry, I realized I hit the names, but the avatars are still a little bit. Sorry. Anyway, okay, so we generate a lot of data, but the data is also not necessarily the friendliest data to deal with as well. Because we're making measurements at the molecular level, we often find that the data is really noisy, right? Because you're measuring things at such a small scale that a little bit of noise sort of really distorts your signal to noise ratio. It's often very sparse because we're detecting so many features that, despite the nature of that, a lot of them will be forced to be zero. The data is often very multi-collinear because everything's just correlated with everything else. And we have a lot of batch and technical effects as well. And just to give an example, so this is some data collected on mass spectrometry. And on the x-axis is like the run order. So like we're running like 1,000 something samples on this machine. And the y-axis is the signal. And you just sort of look at the signal over time, you can see that there's like this really clear trend, which is going to be completely unrelated to biology. And that's because this sort of technology is like really sensitive to like environmental conditions, and you have like signal drift over time where the signal sort of gets weaker, and then when you like sort of clean the machine, suddenly like the signal just jumps back up again. And so we have like really challenging data to deal with. But that sort of brings us to the whole field of like bioinformatics. So it's a really, really exciting field where, given that there's so much data and that there's so many challenges with this data, it's how can we, actually get some meaningful insights out of it. So that's sort of the whole field of bioinformatics. And there's a lot of fantastic research done by the Senior Precision Data Science Center, who create a lot of these software and statistical tools to help analyze this sort of data, and as well as in many other research groups. So this is just sort of like some general challenges about sort of this sort of omics data and sort of bioinformatics in general. But to talk a little bit about the challenges that I will specifically address sort of with the research in my PhD is something like, how can we translate the research findings into the clinic? So a lot of research sort of gets done, collected from these sorts of technologies and this sort of data, but it's not immediately applicable in a clinical setting because there's many barriers in the way. So this can include things like regulations on, how accurate the tests need to be, thinking about monetary costs as well, because if you want to implement something in the clinic, it needs to be affordable and scalable. And also thinking about time constraints as well. And another major challenge in this is that if you do want to bring these sort of findings into clinical practice, how can you ensure that the findings are actually beneficial for everyone? And because you often find that the sort of data that we have, it can be quite biased. And so any model that you train on that data could be biased as well. And you wouldn't want to, implement like a biased model into clinical practice. And to give an example of that, so this plot shows the amount of effectively genetics data. So this is individuals in genome-wide association studies. And over time, and it shows the proportions of different ancestries in this data. And you can sort of see by looking over time, the data has predominantly been on European ancestry individuals. And that's not very surprising because, this sort of research is very expensive to do. And so typically, it's, countries with European ancestries that have had the funding and resources to be able to collect this sort of data. And this is compared to like, on the right here, you've got the actual global population and the proportions. And you can see that the data that we have is not very representative of the global population. And so that's sort of the data that we have. And so it's not surprising that when you look at the actual prediction accuracy of these models on these different ancestries, so here this is plotting the prediction accuracies relative to European ancestry. You can see that the models will perform worse relative to the European ancestry. So, considering that, the data that we have might be biased, how, if you are going to implement this into the clinic, how can you make sure that, what you're implementing is, is beneficial for everyone and not even like exacerbating social inequalities? And so that brings us to sort of the first project I want to talk about, which is multi-platform clinical pathways. Going for time. Oh, okay. Right. So for the multi-platform clinical pathways, to sort of motivate this idea, let's just sort of imagine that you're a doctor. So let's say you're a skin doctor, and you have patients that sort of come in with a spot on their skin, and they want to check, is it cancerous or not? Okay. So so let's say you're a doctor, you have a patient called Jamie who walks in, and from your experience, you can sort of look at the spot, and maybe you can say, oh, no, this is okay, Jamie, you're right, you're good to go. No need to worry about that. Yeah, nice and easy. But then maybe on the same day, someone else walks in, Jackson comes in with another spot on his skin. He comes in, but maybe you have a look. Oh, but this time you're actually not too sure, right? So because you're not too sure, then maybe you sort of get a biopsy and then you send it to the lab and get some histopathology done. And then maybe based off the results of those, then maybe the result is clear. Maybe yes, it is cancerous, and we need to do something with Jackson. Okay, but then let's say another person comes in, maybe Martin comes in, and again with a spot on his skin, and maybe again you have a look, you're not too sure, so you take a biopsy, send it to the lab, but then maybe the lab gets back and and the result is also inconclusive. So then maybe you go do some more tests, do some sequencing, maybe it's still inconclusive, maybe then you maybe you need to do some genetics analysis before you can finally get a confident diagnosis. So what you can see here is an example of a clinical pathway. And given that nowadays doctors have so many different tests to pick from, the million-dollar question is, well, how do you actually construct such a pathway, right? So if you have a lot of patients coming in, how can you decide what tests to do in what order to make a confident diagnosis? And when you're trying to construct this pathway, you also need to think of it in terms of not only being accurate, but also being cost effective and possibly time effective as well. And so that brings us to our framework, multi-P, which stands for multi-platform precision pathways, where we actually build these clinical pathways from data, so that way we can sort of build these in an unbiased, quantitative way, and we can also optimize these on accuracy and cost. And so to explain how we do that, so the setup is that you have data from a few different platforms. So just for this example, let's suppose we've got 4 different platforms, and what we want to do is we want to perform multi-stage classification. So the idea is you've got your whole cohort of patients. So this is maybe all the patients that you'll see in a year, for example. And you can imagine that the first thing you do is you'll look at your cheapest platform, using the clinical data available to you. And from there, you might be able to make a confident diagnosis. Maybe they'll be good. Maybe they'll have a poor outcome, or maybe you're uncertain. And that's when you sort of bring them to sort of the next stage, where you sort of get another test, and then from those results, you might decide if they are they have a good outcome or poor outcome, or if you need to do another test, and that sort of continues on. So that's what we're going to try to build. You can sort of see to do this, the key thing is to consider an uncertain class. So this is sort of the new thing on top of the traditional classification framework, which a binary classification framework, where you typically try to classify people into one of two classes. And here that'll be like a disease positive or negative class. And the way we do that is that we're going to use a confidence threshold to sort of decide whether we classify someone in a particular platform or whether we sort of pass them to the next one. So the way we sort of do that, so the input into this framework is data from multiple platforms. So this data sort of all corresponds to the same set of samples, so the same individuals. But in your different platforms, you'll have some number of features. So these can be like your different omics platforms or different sorts of tests that you can do. And they might have different numbers of features. And then for each individual, you have some outcome. And so what we do is we first train our classification models. And the way we do that is in each platform, we sort of split our data. And here you don't have to look this way, but we just split it like 50-50 into like two folds. And then we repeat this process like 100 times. So you can almost think of this like a cross-validation framework. And what you do is, on these subsets of the data is you train a classification model. And what this gives us is that it gives us a way to estimate a probability of having the disease. And to sort of illustrate how it works, we can sort of do it in two frameworks. Like you can apply this on a sort of sample in your data set, or you can apply this onto an independent data set. And just to sort of explain how it works is that if you have a sample inside your data set and you want to sort of train these models, we sort of split the data 50-50 and we choose the fold that does not include that sample. So that way you're sort of training a model that's independent to your sample, which you can think of like as your testing data. And so because we repeat this 100 times, you'll have 100 different models which have a prediction for that individual sample. And from those 100 predictions, then you can calculate a probability of having the disease. So you can almost think of this as like if you had like 100 different experts trying to evaluate a patient, and then those 100 experts will each have some binary classification, and you sort of aggregate those together into a probability. But also, if you wanted to apply this to an independent data set, because it'll be an independent data set, we can use the models trained on both folds for all of your repeats. So it's a similar sort of idea where you get 200 predictions and some sort of probability. So in any case, so for each of these platforms, you can calculate a probability of someone having that disease. And then we can deploy a clinical pathway. So what we do is you pick a candidate sequence. So like you pick which platforms you want to use in which order. And then from each of these platforms, you can estimate each person's probability of disease. So that was sort of using this framework from before. And then you can sort of imagine that, okay, so you estimate the probability of disease for each person. Some people will have a high probability of disease, some people have a low probability of disease, and you have some people in the middle. And from that, you can decide whether to make a confident diagnosis at that stage or to pass them on to another platform, right? So if their probability of disease is really high, then you'd say, oh, yes, they have the disease. If the probability is very low, you say, oh, actually, they'll have a good outcome. But if it's something in the middle, then you'd say, well, actually, I'm not too sure. So then I'll go collect more data from another platform. Right? So it's like doing another test. And then you can imagine that you can just sort of repeat this process for all of the platforms that you have. So this is the framework of what we did. And just to give an example of how it performs. So here we're trying to classify coronary artery disease in in the BioHeart CT cohort. And we're using data from 4 platforms. So this includes clinical information and 3 omics platforms. And here, this shows the results of a few different types of models. So on the x-axis, we have the total number of assays collected, which you can think of as like the number of tests that need to be done, right? Which is sort of like a proxy for how expensive the pathway would be. And then on the y-axis, you have the balanced accuracy, so like how well the model performs. And so in blue, these are the platforms where if we just use a single platform, and then we have to make the decision, this is what we would get. And you can see that it's towards the left, which means it's like quite cheap, but it also has quite low accuracy, right? So if you only use information from one platform, you know, your predictions are cheap, but not necessarily the best. And then over here on the right, we have the combined ones. So this is if you use all of the data available to you, right? So you can see if you use all the data available to you, it's the most expensive one. You have to collect a lot of data. And you can see, you know, it performs slightly better than if you use a single one, but it actually doesn't perform that much better. And that's because this data would be really noisy. So if you add too much noise into it, it can actually lower the performance. But then in green, these are the ones where we use our constructed clinical pathways. And what you can see is that these green ones, they do like a little bit more tests than these blue ones. And that's because, you know, we're sometimes passing on people to the later platforms. But it's not actually using that much more, right? So it's still a lot less compared to using all of them, which means that we don't need to use all of those tests. But then you can see that the accuracy is a bit higher than all of the other ones. So what you can see is that, you know, when you use this framework to construct these platforms, when you use this framework to construct the pathways, it improves the accuracy with limited increase in cost. Okay. And so as a part of this project, we also came up with a lot of different visualizations and ways of summarizing these pathways. So here, this is just sort of a flow chart that shows what proportion of the population sort of gets classified at each stage. And here, this is just a more detailed look at that, but I won't go through that in the interest of time. We can also compare a few different candidate pathways. So here, these are a few different pathways that we're testing, which is like looking at different orders of the omics platform. And then we also have a visual comparison as well. So here, these were 6 possible pathways corresponding to like a few different orders of the platforms. And we're plotting the accuracy against the total cost. So you can sort of see the ideal place is to be in the bottom right, because you want to be more accurate, but with a lower cost. But what you can see is that often it ends up being like a trade-off between how accurate you want to be versus how much you're willing to spend. Right? You have to spend more to get more accurate, models. And then just as another thing, we also show that that these pathways can also be transferable between one cohort to another cohort. So as another case study, we looked at the prognosis of stage 3 melanoma, and then we trained on data from the Cancer Genome Atlas, and then we applied those pathways onto data from the Melanoma Institute of Australia. And then these were the platforms we used, and then we find that when we build the platforms on one data set and apply it on the other data set, it sort of maintains an acceptable performance. And then just one final thing about these pathways is that we also show that by using these clinical pathways, we can actually ensure that we have fair predictions, even if your individual models are biased. And to show how this works, you can imagine like a context where we have 2 platforms, and you can imagine one of them is like a cheap platform with like a decent accuracy, and another platform is a bit more expensive, but it's like your more reliable test. And this is actually quite a common scenario where you have like a really cheap like screening tool and like a more accurate tool afterwards. And I think this even happened like during COVID, like where you had like the rat tests, which are sort of really cheap and fast to do, which gives you a result, but they're not like the most reliable thing, but has like okay accuracy. But then you also had the PCR tests, which were way more reliable, but a little bit more expensive and time consuming. So it's a so it's a common sort of scenario. And then because we're talking about fairness, let's suppose that these platforms perform better for one group over another group, right? So that we have these bias classifiers, so they always do better for one group. So what we can do is we can consider the trade-off between cost and accuracy for each of these groups. So the idea is that when you have this cheap platform and this expensive platform, what you'll do is with this cheap platform, you sort of make everyone do that first because it's like your screening tool. And then based off the results of those, only if you're uncertain, then you would sort of pass them on to the more expensive platform. And so what you have is that you have this sort of threshold that you can choose to alter to decide, whether or not to pass people onto this second, more expensive platform. And so then we can construct these this trade-off plot. So here we're pretending the two groups are let's say like male and female, because there is a lot of unfairness between males and females in medicine and like in medical treatments. And so for each of these groups, what we're plotting here is if you use a different confidence threshold, what the trade-off would be between error and cost. So for example, on the top left, this is what happened if you used a lower confidence threshold. So what that means is that you are mostly classifying people with your cheap platform, right? So what that means is that you're mostly using this sort of cheaper, less accurate platform. So that's why you have a lower cost but higher error. Whereas if we had a scenario where we had a higher confidence threshold, so you're passing on more people onto this expensive platform, you can see that it would cost more, but then it would generally be more accurate. And then these gray lines correspond to what the result would be if you use the same threshold for both groups. And what you can see is that for the female group in this situation, it's generally like higher and to the right, which means that if you use the same threshold for both groups, the platform would be more accurate for the males and less accurate for the females. And what we show is that, well, if you have this choice of two platforms and this sort of confidence threshold, what you can do is actually use a higher confidence threshold for your disadvantaged group to enforce more accurate predictions, right? So you can sort of think of it as, well, even if your classifiers are biased, we can spend more money to get more accurate predictions for our disadvantaged group. And that's sort of what's represented by these purple dots here. So by using different confidence thresholds, you can see that they'll have a similar classification error, but it'll be spending more money for the disadvantaged group. And to just sort of explain how we do this, is that you can sort of imagine that what we're trying to optimize is sort of a few possible things. So you can sort of imagine that if the purple things are your optimal thresholds, and sort of green is sort of like the centroid of that, you can imagine that we might be interested in controlling for the average error of the whole population, the average cost of the pathways on the whole population, or the difference in error between the two groups. And so then what we have is that because there's sort of these three things that we're trying to minimize, what we can do is you can fix two of these things. And because you've got sort of two thresholds to play with for both groups, you can sort of find a unique minimum for the third one. And And that's sort of how you can sort of enforce that minimum. And then we also deploy this in a Shiny app so that you can upload your own data, or you can use simulated data and sort of see how this trade-off looks. Okay, and just to wrap up this part of the presentation, so there's still a lot of scope for future work. So for example, in the pathways that we make, we sort of enforce the same pathway on everyone in the population. And so there's scope of doing more personalization in the pathways by looking at like different orderings, using different classifications or different subcohorts. And then also studying fairness in a general setting. Because in our case, we only have 2 platforms and 2 groups, which is a very simplistic situation. So extending this into a more general setting would be very interesting. And our paper is also published in scientific reports last year. Okay, how am I going for time? How much time do I have? Is that 10 minutes or is that no more minutes? Okay. Okay, excellent. Okay. Okay, so in the last 10 minutes, I'll briefly talk about another project that I did. Yeah, which is sort of a fun little project, looking at lipidomic signatures of resilience to coronary artery disease learned from chimpanzees. And what on earth is that about? Okay, so to sort of motivate this project, I'll talk a bit about a story of 2 individuals. So one of them we'll call Andy, and another one we'll call Sam. So Andy is a human, and Sam is a chimpanzee. So as a human, Andy is actually quite likely to die from coronary artery disease. And in fact, coronary artery disease, it's the leading cause of death in Australia and in the world. But Sam, as a chimpanzee, is actually very unlikely to die from coronary artery disease. And in fact, there's only been a single report of a death from coronary artery disease. And this is true despite like thousands of necroscopies having been done. And this is really surprising because humans and chimpanzees have like a really big overlap in DNA, like they're our closest animal relative. And that leads us to the questions of, well, why are chimpanzees so resilient to coronary artery disease? And what can we learn about human coronary artery disease by studying this difference? So to do that, we sort of embarked on this project where we looked at a cohort of chimpanzee volunteers and some human volunteers. And I have to do a big shout out to my collaborators, Michael and Gemma at the Collins Institute for sort of managing the cohort side of things. And then we collected lipid data from the chimpanzees, as well as lipid CCTA imaging, which is looking at coronary artery disease, and genotyping data from the humans as well. And also a huge shout out to our collaborators at the Baker down in Melbourne for doing all of the lipid mass spectrometry for us. And in case you're not familiar, so the reason why we're looking at lipids so specifically, or like what lipids are, is that in general, they're just a category of molecules. And the defining feature about them is that they're insoluble in water. But the reason why they're interesting is because they have a lot of really important biological functions. So including like molecular signaling, energy storage, and they form a big part of cell membranes. And so for a complex disease like coronary artery disease, it's really interesting to look at sort of the lipidomic profile of these cohorts. And then we do a bit of analysis to try look at the differences and the causes of these differences and link that back to coronary artery disease. And so with this sort of cohort, we tried to answer a few research questions. So naturally, the first thing is, we have these chimps and humans. It's sort of, well, what's different between them? And then thinking, okay, so once we look at what's different between them, how is that related to someone's resilience to coronary artery disease? And then seeing if we can link it back to genetics, because we're looking at chimpanzees, which is quite interesting. Okay, So let's have a look at these. So for the first one, looking at what is similar or different between humans and chimpanzees. So the way we do that, so I'll walk through our approach to this. So we start off with 768 lipids, but these lipids fall into different categories, which are sort of related to their function. And we're often more interested in looking at how these classes are sort of different between the two groups. So what we do is you can imagine that for each lipid, so each individual lipid, we can sort of do a comparison between chimpanzees and humans, right? So it's like essentially just like a t-test. And then we can rank all of the lipids by test statistic. So this is all 768 lipids. And then what we'll be interested in seeing is whether these classes are generally higher in humans or higher in chimps. And so that's what we do with in this lipid set enrichment analysis. And so that way, you can see that if a lipid class is generally more towards a positive end, it would have a lower abundance in chimps or higher in human. Whereas if it's on the negative end, it would be higher in chimps. So we run this across all the lipids and all of the classes. And in the interest of time, I'll just quickly skip through this. But we sort of had a look at the properties of the lipids that were particularly high in chimps or particularly high in humans. So here, this is like a bar plot of the human average minus the chimp average, like lipid concentration. So if it's like below the x-axis, it means it was higher in chimps. And if it's above the x-axis, it means it was higher in humans. And so these are examples of some lipids that were very high in chimps. And this included things like omega-3 and polyunsaturated fatty acids, which are known to be very healthy. Whereas the things that were higher in humans were, I won't go into this, but some chains which might be bad. And so then we also looked at classes of lipids. So some things that were higher in chimpanzees included these types of lipids that were, which were like, had very strong antioxidant properties, as well as these signaling molecules for important lipid pathways. And if we look at some lipids that were particularly high in humans, we also saw these interesting lipid classes as well. Okay, and so that was comparing the lipid profiles between humans and chimps. And then we also wanted to look at this lipid profile and its association with coronary artery disease. So here you can sort of imagine that if you look at all of the lipids, you can almost think of it as defining like 768 dimensional space. And the humans and chimps, there's like some point in this space. Right here, I'm just sort of visualizing it on a PCA plot. But then your hypothesis might be that, well, if chimps are so protected against coronary artery disease, are the humans that are closer to the chimps, would they also be protected against coronary artery disease? And that's effectively what we set out to do. And so just to quickly explain how we did that, so with the chimpanzees, we calculated like the average of those, and then we calculated the distance between each human to this chimpanzee average, and then we had a look at the association between this distance and coronary artery disease, and pretty much it had a very strong association, even when you control for like all other known risk factors. And then we could also binarize this. So looking at like a chimp signature, so just taking like the 25% of humans closest to this average, and then if you compare them to the people who are sort of far away from this average, and we look at their coronary artery disease outcomes. So here, this CACS, Gensini, and soft plaque score are like different types of outcomes for coronary artery disease. And what we found was that the people who were closer to this chimp signature average were much healthier compared to those who were further away. which is quite interesting. And then a final thing is looking at the genetics. So seeing what, so seeing if the differences between coronary artery disease and these differences that we observe in lipids, could they be related to evolution? And so I will just sort of quickly skim over the analysis. But the idea is that we are trying to find lipid SNP associations. So you can think of these as like genetic lipid associations. And that sort of satisfies certain categories. And to give you some context, we have about 768 lipids and about 14 million SNPs. So there's a lot to start with, but we're trying to find the ones which are, firstly, like we have evidence to say that they're associated, and we sort of combine some data sets to do that. We want the SNPs to be associated with coronary artery disease. So we have to use information from another data set. And then we also look for SNPs that are under positive selection pressure. So that would be affected by evolution. So we use another data set to help us with that. And then we look for ones where the effect that the genetics has on the lipid matches the comparison between humans and chimps. And then we find then we filter to those where the lipid abundance is associated with coronary artery. And so that was our workflow. And then we found these 5 SNPs, which were not located, but they were mapped to some really interesting genes. And so these are quite well-known genes for coronary artery disease, and the lipid classes that they were associated with were also quite interesting because they were some of the ones that we saw earlier on as well. Okay, and so just to quickly wrap up, so with sort of these 3 research questions, we sort of had some findings. So that we saw that the lipid classes in the chimp profile were generally like quite good, and the ones that were higher in humans were generally quite bad. And then we also saw that the humans who have a similar lipid profile to chimps were generally more resilient to coronary artery disease. And then we also saw that some of the genetic differences between humans and chimps might be contributing to the differences in coronary artery disease. And so here, this was just looking at the lipids, but coronary artery disease is a really complex disease. And this was actually just like the start of an even bigger project where we want to look at many other omics as well in chimpanzees compared to humans. And so that's all I wanted to share in terms of my PhD research. So this is a really, really exciting field of precision medicine and bioinformatics. But hopefully, my PhD research is like a drop in the ocean, contributing to that. And to wrap up, I really want to thank all of my amazing collaborators, because this sort of research is very, very interdisciplinary. And so it definitely could not be done without, help from these really wonderful people, especially these wonderful people here in the Sydney Precision Data Science Centre.